Hi, this is Dr. Dan Guerra coming to you from VerEvMed and Authentic Biochemistry Studios in the Pacific Northwest of the USA. Today is actually the 29th of August, 2019. And what I'm going to cover today is we're going to try to finally carry out that last interrogation of T regulatory lymphocytes, summarize what we've been discussing from the last video lecture, which was earlier this week, plus the 6 segments I did on Authentic Biochemistry podcast, where we went through the entire T-cell modulatory theories, went through a lot of data, discussed several research papers, where we focused primarily on how T regulatory cells can be used to dampen down inflammation, as well as to modulate the TH lineage in a way that can control and perhaps ablate tumors. So this is all a huge biomedical interest that's been going on now for at least 10 years. So there are several immunomodulating pharmaceuticals that are out there that help regulate T-cell lineages. And we went through several of those. And rather than discuss this from a pharmaceutical perspective, I've still been staying primarily in the physiological and pathophysiological modes and how they relate to disease, to potentially just regular nutritional and dietary states relative to a healthy lifestyle, and then how that may have ameliorative effects in the disease state itself. We haven't really talked much about age or gender relative to T-cell modulation, but I'm going to try to get into that a little bit today. So without any more discussion as a prolegomena, let's get started. So, as you can see here, this is my Facebook page, and this is my podcast link. I'm Dr. Daniel Guerra. This is my email address. You can contact me about this and any of the VerEvMed lectures you see online. And in fact, you can contact me about any of my podcast lectures, which are on authentic biochemistry, which is found in all the regular platforms for podcasts. I'm not going to talk to you today about my company, VerEvMed, or about authentic biochemistry. We've done that before, and we'll do that again later. Right now, I just want to jump right into this discussion. So today's topic is T regulatory cell logistics and a guide to the Diaeventontology for pharmaceutical manipulation of acquired immune response, which of course T cells are the major player along with B cells. So now this diagram here comes from the same paper we've been looking at in the last series of slides. And what I want you to see is that you've got these various subclasses of T cells, starting from a naive CD4 positive T cell, interacting with some kind of antigen presenting cell. This is showing here like a dendritic cell, but it could be a macrophage. And you can see here that, and I've written this down, and you can also see if you follow the diagram, the Th1 lineages deal with intracellular pathogens. They work with macrophages. They involve inflammation. Th2 lineage work through these cytokines, interleukin 4, 5, and 13. And they are, Th2 cells mount a defense against parasitic helminths. They interact with mast cells, eosinophils, and they're often associated with allergy. The same CD4 T cell lineage, if it's not induced by IL-12 and 18, become a Th1. And via T-beta is the transcription factor generating interferon gamma and doing this business here in terms of immune responses. And if it's not under the influence of interleukin 4 and using the transcription factors here and the signal transduction cascade of the STAT system, working through IL-4, 5, and 13 with that system, you can also take naive CD4 plus T cells, and in the presence of TGF-beta, turn on either Th17 cells. Th17, the major transcription factor there, is the retinoic acid orphan receptor called gamma T. Okay, so that is what's expressed to make Th17 have that particular molecular and cellular signature. So interleukin 23 and the intracellular phosphorylation cascade system of STAT3 will then give you mature Th17 cells. They'll work with extracellular bacteria, fungi. They also recruit and interact with neutrophils, and they are often associated with autoimmune disease. Now, the same naive CD4 T cell, finally, in the influence of TGF-beta, and then utilizing the SMAD system, the phosphorylated SMAD system, interleukin-2 being another important ligand to bind to a receptor to go down this pathway, working through the STAT5 system intracellularly, you get the expression of FOXP3 transcription factor, which then turns on all the genes necessary to be a Treg cell. Treg cells make more TGF-beta, which can allow them to make more themselves, but they also make the immunosuppressive interleukin-10. Remember, this causes suppression of the immune response, which we talked about, and the other term for that is tolerance. So I think I've said all this. Upon antigen stimulation, CD4 T cells follow distinct developmental pathways. We looked at Th1 through interferon gamma, Th3 through these cytokines. Remember, cytokines are glycoproteins that bind to receptors that cause modifications in gene expression in the cell type that they have been inducing. That's what cytokines do. They can be pro-inflammatory and anti-inflammatory. Immune pathogenesis that results from dysregulated Th1, by the way, to either the self or to some commensal floral antigen, can promote tissue destruction and even chronic inflammation or even autoinflammatory responses. Whereas a dysregulated Th2 cell lineage can also cause allergy to compounds that should not be allergens, and is also associated with asthma. Th17 secretes a distinctive set of immunoregulatory cytokines, as we just said, IL-17, IL-22 in particular, of course, from Th17 lineage. They also play a role in inflammation and also a big role in autoimmunity. TGF-beta also induces differentiation of those naive T cells, again, to FOXP3, transcription factor expressing Treg cells. Okay. Now, before we continue, this is a little bit of fun here because you remember I'm a biochemistry professor. And so as a biochem professor, you go through pathway after pathway and you make all kinds of really interesting discussions and you demystify otherwise mysterious pathways, particularly for medical students, vet science students, even graduate students who think that these molecular pathways just looks like a lot of unknown systems leading nowhere. Of course, that's not the case. In fact, indeed, they're all interconnected. So before I get into the next synoptic discussion of T cells, I want you to think about a couple of things that are necessary for T cells to work. From the diet, essentials are niacin, and also tryptophan. So this is tryptophan up here, and that's niacin down there. Okay, these are both nucleotides, by the way. That is, they have heterocyclic ring structures, or they have, at least in this system here, tryptophan is utilized actually in nucleotide biosynthesis. Anyway, L-tryptophan is an essential amino acid. It's converted to L-kynurenine when then upon reduction and in the presence of FAD and two other vitamins, 6 and 2, will make this quinolinic acid. And then the presence of phosphoriboside pyrophosphate and magnesium divalent cation, and then adding the niacin, you make the dinucleotide. You can't see it down here, NAD, nicotinamide adenine dinucleotide, which of course is the major redox player, and also the reduced form of it is how you get proton translocating across the intermitochondrial membrane through the electron transport chain, relative levels of oxidation of reduction of molecular oxygen down to water, and then the movement of those protons back through the proton pumping complex 5 ATPase in the intermitochondrial membrane, making ATP. Okay, I want you to keep in mind though, you got tryptophan and you've got niacin, which are essential in the diet to do this. Okay, now check this out. Not only them, but also vitamin A. So retinoic acid, is a vitamin A metabolite. It's produced in the gut. And it's in fact produced in the gut-associated lymphoid tissues, which are called GALT. So there are CD103 plus dendritic cells in this tissue. And they induce the conversion of naive CD4 plus T cells to Tregs. And that's what's shown here. Okay, so these dendritic cells, which are in the GALT complex in the human gut, take the naive cells and turn them into FOXP3 expressing T regulatory cells. They require vitamin D metabolite 1,25-dihydroxy vitamin D3, which can be synthesized via exposure to UV light in the skin. So it's actually not a vitamin. It's sort of a vitamin if you are deficient in having enough sunlight. Vitamin B3, which we already said was niacin, and vitamin A. Okay, so here we show you how all of these come together to interact with their receptors. There's a GPR 109. So vitamin B3 binds to the G protein couple receptor, okay, binds to the dendritic cell, and other innate immune cells, allowing those cells to induce the FOXP3 transcription. We already talked in great length about FOXP3 transcription. There are 3 different enhancer elements in its promoter, all of which are heavily modulated by a whole slew of transcription factors and adapter molecules. So vitamin D3, through its metabolite, works through its VDR, which is a vitamin D receptor, of course, enhances FOXP3 expression, making that mature T regulatory cell. And vitamin B9 metabolite, which is, of course, tetrahydrofolate. That's what this is here, THF. That's necessary also for survival of the T regulatory cells. So you have induction of Treg cells from the naive CD4 positive lineage. Then you have to maintain the survival of Treg cells, and that's maintained by tetrahydrofolate. Now, an interesting thing also, notice this. Leptin, which is an adipokine produced by the adipocyte, which normally works in the central nervous system to alert for satiety. So adipose tissue generates leptin. Leptin is translocated to the brain, and it shuts off the gustatory responses and the appetitive responses to eat more. We have all probably heard of the leptin deficient mice that are multiple obese, right? Well, leptin does other things, of course. Leptin is basically a hormone, right? And so when it's generated from the adipocyte, leptin can actually block FOXP3 expression, which does that, it cuts down on T regulatory cell activation. So the more adipocyte, particularly if adipocytes are going through an immune response, because they can, because there are resident immune systems there, and there's also reactive oxygen, can modify the repertoire of circulating T cells in such a way as to diminish the amount of T regulatory cells, which would then enhance autoinflammation. This is one way that obesity is associated with an inflammatory system. Okay? Leptin is produced by the adipocyte, blocks FOXP3 expression, but also the Treg cell itself produces leptin. Yeah, it's not just produced in the adipocyte. And it shuts off its own level of FOXP3 expression and therefore limits the amount of Treg. You can't have a lot of immunosuppression when you need to have the T helper cells fighting off bacteria, fungi, virus, and things like that, and just foreign antigen. Right? So that's the point about all this. This is from Trends in Immunology. That last slide was too, and this was published back in 2015. All right, so move myself out of the way again, sorry. Now, this discusses now back to the gut. This has to do with commensal microbial metabolism, which controls also. So we looked at the adipose tissue controlling Tregs. Now I'm telling you that commensal bacteria within the gut, in association with GALT, also regulates Treg turnover and survival. Okay, so colonization of mice with Clostridia, which would normally be a pathogenic organism, from both mouse and human feces. This is how this experiment was done in this paper. Increased Treg abundance in the colon. Now you might say, well, wait a minute, this is a pathogenic organism. Precisely. In order for Clostridia to survive in the gut, it has to suppress the suppressing T regulatory cell activation. Right? If it does that, right, then it will, if it activates it, if it turns on T regulatory cells, if it increases T regulatory cells, it suppresses the inflammatory response of Th cells. That's what I mean to say. All right, so it's produced through bacterial fermentation. So what does the bacteria produce? Okay, well, that's an important issue here. It takes fiber and it makes short-chain fatty acids. And these short-chain fatty acids like propionate, acetate's actually considered a volatile fatty acid, and butyrate, okay, so C3, C2, C4, very short chain, is typically considered volatile fatty acids, all can interact in such a way as to control T regulatory cell lineages. And this is all made from Clostridia via fermentation of fiber. So the Clostridia stimulate conventional T cells to produce interleukin-2. You need some interleukin-2 to activate the naive CD4 plus cells. There it is there, the interleukin-2, so that you can turn on FOXP3 expression and turn the Treg cell into an active system, right? So the Clostridia is working to make more Treg, so it suppresses the immune response where the Clostridia is trying to reproduce. Okay, so not only that, you get B. fragilis, which is another organism in the gut. It produces polysaccharide A or PSA, so shown here. That can induce Treg induction as well, and it potentiates Tregs to produce the regulatory cytokine IL-10, which is suppressing cytokine. In a feedback loop, Tregs contribute to diversification of Clostridia species by promoting the generation of a selection of IgAs. So some of the IgA will control Clostridial species diversification, so that only some species of Clostridia are found in the gut. So that's basically what's going on here. You have receptors for all this. You have GPR43, which is particularly associated with these short-chain fatty acids. Now look, this short-chain fatty acids also inhibit HDAC. HDAC is histone deacetylase. Now, if you inhibit the deacetylase, you maintain the acetylation of the histones. Maintaining the acetylation of the histones allows for the 180 turn from heterochromatin to euchromatin, so the genes are expressed in a facile manner. So if you inhibit deacetylation, you allow for the acetylated histones to open up double-stranded DNA, unravel it from its histone core complex, and thus allow for transcription of genes. Which ones? The ones where there are promoters and transcription factors that are going to turn on whatever cell system you've been activating. In this particular cell system has to be Tregs in the gut. Okay, so that's how it all comes together, right? There's PSA working, and you have toll-like receptors also involved on these naive cells and even in mature Treg cells, and they're all interacting with this PSA, again, come from B. fragilis. So all of this helps trigger an abundance of T regulatory cells, which basically maintains the microbiome in the gut, so it's not constantly under inflammation. Now, in inflammatory bowel disease, this gets dysregulated, so that Treg cells are decreased in their activation and decreased in their survival, because this no longer functions the way you see it here, which means that there's an inflammatory bowel problem. And all of that can lead to an entire change of the infection court, that is the microbiome in the gut. And when that happens, you can get ulcerative colitis and Crohn's disease. So that's another whole element of this discussion. How T regulatory cells maintain its homeostasis in the gut to prevent autoinflammatory responses, but at the same time, they maintain a specific species diversification of bacteria in the gut and other microbes as well. Now, Mtor, which is a major global transcription regulator in the cytoplasm, it's actually a big complex which controls signal transduction cascade. It's basically when mTOR complexes turn on, called mTORC, you get a global expansion of transcription. So mTOR signaling in Tregs and conventional T cells. So in A, you've got in Tregs, okay, these are Treg cells, the T cell receptor, which remember, it is turned on by recombination events, and the interleukin-2, and so you need TCR activation, and that's usually induced by an antigen-presenting cell through MH2 complexes with that antigen, presenting it to the T cell receptor, and then in association with interleukin-2 receptor-mediated co-activation, the Treg cell comes on. CD36 receptors are also in Treg cells, and they're allowing in fatty acids. So that's an orphan receptor which allows for the flow of fatty acids in for beta oxidation. So you also get the expression of PPAR gamma, which is a transcription factor which turns on beta oxidation. So beta oxidation of fatty acids that are coming into the T regulatory cells, all of that in conjunction with mTORC turning on the de novo mevalonic acid pathway. Now, I know all of you know what that is, but I'm just going to reiterate it because I'm a lipid biochemist, and that is the de novo prenolipid pathway. Mevalonic acid biosynthesis has to do with the synthesis of cholesterol ultimately. And the rate-limiting enzyme in that pathway is HMG-CoA reductase, which of course is the target for statins. So if you have low levels of mevalonic acid, which is what would happen with high levels of statin in the presence of T regulatory cells, what do you think will happen? It'll decrease the amount of T regulatory cells over time. Because it's another thing to consider with people on statins throughout their life, that you're modulating T regulatory cells because you're modulating mevalonic acid biosynthesis, because you're inhibiting HMG-CoA reductase, which is the enzymatic machinery prior to mevalonic acid. And on the way to making farnesyl and geranyl pyrophosphate, and farnesyl pyrophosphate and geranyl pyrophosphate, those prenolipids are actually used as covalent modifiers of most of the receptors on the plasma membrane. Okay. You're inhibiting all of that when you use statins. Bring that up a lot of times because I want you to consider it. So there's where mevalonic acid. That's only in Treg cells you turn on mevalonic acid. It's not going to happen in the other T cell lineages, which we're about ready to discuss here. Okay. Now, that which is vascular adipose tissue associated Tregs, right, exhibit increased expression of PPAR gamma, which promotes fatty acid metabolism and hence stimulates the accumulation in the adipose now. Understand, these are in the adipose as well as in the gut and in circulation and in lots of other resident tissues. What are the ones? Like liver and kidney. Okay. Anyway, you promote fatty acid beta oxidation with PPAR gamma, which is a transcription factor, turns on the beta oxidation genes. And it stimulates the accumulation and suppressive phenotype of Tregs residing in the adipose, which is what you want. You want to have inflammation suppressed in the Treg, in the adipose tissue. And you get that by activation and survival of the T regulatory cell lineages. And this is how it's turned on. Okay, utilization of fatty acids, TCR, there's a T cell receptor being activated by angiopoietin, and then the interleukin-2, and remember, interleukin-2 is generated by other T cell lineages, faithfully committing the interleukin-2 to bind its receptor and turn on the T regulatory cells, which are resident in the adipose. Full stop. Okay, that's how you get Treg suppressive activity in the adipose. Now, down here in B are conventional T cells. You're not looking at interleukin-2 binding, you're looking at binding CD28 that has its own ligand to bind to, that's a co-adapter to the T cell receptor. What you turn on here, you also turn on the mTOR pathway, but here mTORC1 turns on HIF1 alpha, which turns on glycolysis. So rather than oxidation of fatty acids, you see, you're using glucose. So many cells will decide the fate of their downstream transcription factor lineage based on what kind of carbon they're oxidizing, whether basically it's glucose or fatty acid. Also amino acids are the other third, sometimes very important player, particularly in tumor cells, by the way, something I've covered before. But at any rate, in your regular T cells, you've got glycolysis, and glycolysis can inhibit T regulatory cell stimulation, okay? And so that's one thing that happens through the mTOR pathway. Notice it's different because you're not turning on the mevalonic acid pathway here, right? Now, even in the Treg cells, mTOR2 will, now look at this here, block Treg suppressive activity. So this is a feedback controlling mechanism. mTORC1 turns on mTORC2. It's just a different complex of the target of rapamycin. That's what TOR stands for. C stands for complex. So there's always a suppression of a suppressor lineage to maintain a correct level, you see. But here in regular T cells, you're inhibiting FOXP3 expression. Here you turned on FOXP3 expression. PPAR gamma also does that. Remember, FOXP3 is the global transcription factor for what? T regulatory cells. Here you're inhibiting it, no FOXP3. That might be regulated by glycolysis, and the way that I'll show you in a minute. And so you get a decreased amount of T regulatory cells. Okay. What you do get in regular T cell lineages with mTORC1 and 2 is a turn on of Th1, Th2, Th17, and the T follicular helper cells, which is yet another lineage. And I talked about that in the 6 segment episodes of Authentic Biochemistry, which you go back and look at. So, but you also make some CD8 memory cells this way. Actually, you block it with mTORC1 and you turn it on via this effector CD8. So again, it's regulatory. So some memory cells are being made from the TH lineage. Okay. Next slide. All right. Now, it's a paper published in Frontiers of Immunology up here. You see, published in 2018. And the title is Immunometabolism of Phosphatidylinositol 3 kinase signaling as a link between interleukin-2 T regulatory cells, FOXP3 T regulatory cells expression, and the suppressor function of regulatory T cells. So now we're talking about how you get an immunometabolism functioning through the P1-3 kinase, and that's what's shown here. So pathways promoting glycolysis and fatty acid oxidation in regulatory T cells and known mechanisms affecting FOXP3 is what the topic of this slide is. Glycolysis is primarily activated in Tregs through mTOR, which suppresses FOXP3, and Treg lineage stability and Treg lineage stability. Activation of the P1-3 kinase through the AKT through the mTORC1, it's all that there, right? Move this out of the way. Inhibits FOXO transcription, okay? So that FOXO13 transcription is actually necessary for FOXP3 transcription. Okay, so FOXO13 is not FOXP3. They're two different transcription factor systems, okay? But in order to turn that on, you can't have this pathway functioning. So what this does when it works through the phosphatidylinositol 1,3 kinase pathway, through the AKT, it's all very canonical and signal transduction cascades, all phosphorylation cascades. Through the mTOR, it's going to block FOXO13. You're going to not make then FOXP3. See, you're blocking this so you don't get this pathway. You also, through mTOR, make HIF1 alpha. Okay, that's hypoxia inducible factor 1 alpha. That blocks FOXP3. You also turn on what is GLUT1, that's glucose transporter 1. So that'll promote glycolysis, right? Now, when that happens, that will block this enol-1. It'll disengage enol-1. Now, that will relieve some of the depression of FOXP3 expression, because this enolase, which is an enzyme in glycolysis, is recruited back out of the nucleus into the cytosol to help glycolysis function. Of course, glycolysis needs the enolase enzyme. Now, of course it does. So you see, there's always, when things are turned on, there are always mechanisms within the major upstream modulating pathway to turn on another pathway to allow for some stimulation and some deactivation. That's the way it works in every system. So there's some push and pull in every system. And that's just showing you here. You also make this EZH2, which is a co-transcription factor, and that can actually enhance some of the FOXP3. Remember before I showed you how glycolysis can have a regulatory role in FOXP3 expression? Turns it off and turns it on. I mean, it's amazingly cleverly designed, where the cell cleverly expresses all of these different gene systems in a healthy environment to regulate the amount of Treg cell activation and survival. So that's what all this says here. Under certain conditions, glycolysis promotes FOXP3 expression by disengaging the enolase. I just said that from its nuclear role. Glycolysis also represses the microRNAs such as miR-101, miR-26A, to enable expression of this co-transcription factor to turn on FOXP3. Okay, so it's also working at an epigenetic level because it's working with interfering RNA species, which is epigenetic, right? It doesn't have to do with changes in the nucleotide sequence. It has to do with changes in gene expression by regulating gene expression at the chromatin remodeling level. Okay, by modulating histones, by modulating methylation, acetylation, things like that, of CpG islands for the methylation and acetylation of histones. And indeed other covalent modifications. So Tregs generally rely on fatty acid oxidation. We already said that. In the gut, short-chain fatty acids, we already looked at this, inhibit histone deacetylases to promote FOXP3 expression and conversion of naive CD4 cells to the Tregs. Okay, that's P stands for peripheral. Under certain conditions, fatty acid oxidation may also affect Treg stability. Okay. SIRT1, which is one of those deacetylates, represses FOXP3 either through direct deacetylation of the FOXP3 promoter region or by targeting FOXO transcription factors. And that was what we see here. See? So working through that, the deacetylases can work through that or work directly on inhibiting. If you deacetylate that chromatin structure, you're going to inhibit the amount of FOXP3 because that's the major gene being expressed, so you're going to inhibit it first. In CD8 plus memory cells, cytokines such as interleukin 7 and 15 promote the uptake of fatty acid precursors and increase fatty acid oxidation, respectively. So that's keeping the fatty acids moving through. So both glycolysis and fatty acid oxidation can also promote FOXP3 expression through another transcription factor called NFAT, okay, which promotes FOXP3 expression through the NFAT dependent mechanism. So this can work with cholesterol metabolism, and we talked about that. So you need cholesterol metabolism, that mevalonic acid pathway in Treg cells, so that you then induce fatty acid synthesis. So not only fatty acid synthesis, because the earlier prenolipid accumulation in cholesterol biogenesis. So, for example, past mevalonic acid up to farnesyl and up to geranyl pyrophosphate, those prenolipids help control fatty acid synthesis by controlling acetyl-CoA carboxylase and FAS expression. Yeah. And there's CD8 memory cells turning on IL-15 and 7, working through this carnitine palmitoyl transferase 1A, turning on fatty acid oxidation. You make some reactive oxygen species, and that will turn on NFAT, transcription factor, and that working with CNS2 then will enhance FOXP3 expression. And that, of course, will then turn on the T regulatory cell lineage. So this is how different metabolic pathways, you see, lipid metabolism plays a very critical role here. Fatty acid oxidation and mevalonic acid biosynthesis. So all the acyllipids and all the prenolipid pathways are turned on in FOXP3 expressing T regulatory cells. And it works through the mTOR and the rarefied lineage because of what genes are regulated in Tregs versus what's happening in TH cells. All right. Now in adipose, in visceral white adipose tissue, or WAT, there are regulatory T cells, okay? And they're involved in metabolic homeostasis. So here's your regulatory T cell. It has a chemokine receptor, CCR2. It has this ST2 receptor, okay? And it also has the FOXP3, has the PPAR gamma. These are transcription factors which are going to control all the genes that are going to mediate Treg activation. And this is in a healthy adipose tissue. You also have some granulocytes, you have some Treg cells, you have some M2 macrophages. Again, the story about macrophages, M1, pro-inflammatory. Remember, those are antigen-presenting cells, part of the innate immune response. M2, anti-inflammatory, okay? So that lineage can switch back and forth, M1, M2, M1, M2. So in a healthy adipose tissue, you have plenty of T regulatory cells suppressing inflammation. You have plenty of M2 macrophage lineages suppressing inflammation. Okay. And all of this is working with the extracellular matrix as well. That's necessary to carry out the signal transduction cascades so that it can interact with chemokines and cytokines. Okay. So when you have a sustained high energy balance, which basically means overeating, you change the adipose tissue. See? And what happens is you get M1 macrophages, which are pro-inflammatory, and you get a decrease in the amount of T regulatory cells, a reduction. And that's because you decrease the amount of FOXP3 expression. So WAT Tregs adapt to adipose by expression of PPAR gamma, see there, that regulates genes involved in beta oxidation. These Tregs also express alarmin, interleukin 33 basically, the receptor, which is ST2, okay? That's actually an interleukin 33 is also known as alarmin, okay? And of course the chemokine receptor. All that's what's going on in the adipose to keep those Tregs happy and active. So Tregs are abundant in lean adipose tissue, okay, of adult mouse. These are in mice, okay? However, if there's a sustained positive energy balance, means overeating. So in humans, it's not so much the high-fat diet, it's the high-carbohydrate diet. And we've talked about this many, many times in VerEvMed and in authentic biochemistry. So I'm not going to belabor it here. In mice, they give a high-fat diet, and that's why it was believed the high-fat diet would do the same thing in humans. Actually, a high-fat diet suppresses a lot of this pro-inflammation, unless it's way out of whack. It's the high-carbohydrate diet, which turns on pro-inflammatory responses, and hence obesity-associated metabolic syndrome. And what else? Type 2 diabetes. All right, this is concomitant to a change in macrophage phenotype. It goes back from the anti-inflammatory M2 to the M1. I just said that. See, boom. And you get unhealthy hypertrophy of the adipose tissue. Hypertrophy means it's growing. That's how adipocytes grow. They increase in mass because they accumulate more triacylglycerol and oil droplets. That's how they don't know so much divide as they get larger. That's what adipocytes do. You get unhealthy hypertrophy of the adipocyte. Sustained hypertrophy is how much bigger is supposed to be showing you. The adipocytes themselves are these yellow cells. Overall decrease in adipose Tregs. You decrease the amount of Tregs because you've increased the mass of the adipose, basically. Okay. So it's kind of like a ratio and proportion. Right? You get insulin resistance in the adipose tissue, so glucose uptake becomes inhibited. And also the lipases are deficient. Insulin resistance, dyslipidemia, of course, high levels of triacylglycerol in the serum, and a chronic low-grade inflammation in the adipose, which can lead then to a complete dysregulation of Treg cells, can induce diseases like cardiovascular disease and cancer, because now you completely corrupted the adipose tissue, so it becomes a pro-inflammatory signaling system into the lymphatic system and into the blood, generating a lot of pro-inflammatory cytokines and sending also out a lot of lipids that are binding to receptors and enhancing the amount of lipid accumulation, for example, in the cardiac system, then generating atherosclerotic plaques, for example. So you get the idea. Adipose is really important modulator of the whole body. That's why when you overeat, you get a lot of visceral fat. This is what happens to the T cell lineage. Now, colonic Tregs, back to this. You have three different subtypes. You have the FOXP3 plus ROR gamma T cells. They comprise about 50% of normal colonic Tregs. They're responsible for establishing the tolerance of all the dietary antigens you eat. So, everything you eat has proteins, and those proteins are broken down, and they make limit peptides. Those limit peptides could be antigens. They're not supposed to be. But if you have an inflammatory response, you change the infection court, meaning also you change the commensal bacteria, which are beneficial as a microbiomic beneficial environment in the gut, and you get inflammatory response. This dysregulates everything, and you can get ulcerative colitis, and you can get inflammatory bowel disease because you change the kind of bacteria that live in the gut, producing more reactive oxygen, more pro-inflammatory cytokines, and then there, of course, the inflammatory bowel disorders, right? Yes. So this is basically what you're seeing here. Your food antigens, microbial metabolites, where we talked about these short-chain fatty acids. There's about vitamin D working to make the Treg cells, right? Here's a dendritic cell, decreasing these kinds of cytokines so that you make more FOXP3. The HDAC inhibition because of the short-chain fatty acids, propionic acid, butyric acid, volatile short-chain fatty acid, acetic acid. Microbial antigens working with the Gall system to turn vitamin A into all-trans retinoic acid. That's the ROR gamma T, which you need in the presence of TGF beta to produce more Treg cells. And this is what Treg cells look like. This is their expression pattern, right? And then, of course, you've got these this whole lineage of Th17 cells. So back over to here, vitamin D metabolite 1,25-dihydroxyvitamin D differentiates naive T cells into Tregs by activating its nuclear receptor. FOXP3, ROR gamma T, Tregs deploy various mechanisms toward establishing a dominance and the dominant tolerance motif in the gut. Okay, and they do that with producing a lot of interleukin 10, shown there. Interleukin 10 is going to suppress Th17, which are pro-inflammatory in the gut, right? You see that? It's the way it works. Now, a special subset of FOXP3, ROR gamma T, Tregs express CMAF1 and 2, and it's instrumental in establishing a tolerance to pathobionts in homeostasis. Okay, so that's another component of the cell system. Pathobionts are potentially pathogenic organisms, right, which can survive in the commensal population as long as they don't start dividing, right? So that lineage is also aided and abetted. So FOXP3, ROR gamma T, CMAF plus Tregs are identified against specific epitopes of Helicobacter, which of course It's a major problem, bacterial problem in the gut, and it causes a lot of the diseases that are associated with gut inflammation, right? Because it suppresses the epitope-specific Th17 cells. In the absence of this P-Tregs, early seeding of thymic Tregs, which are T-Tregs, happens in colonic LP. These T-Tregs expand into a niche which is independent of MHC class 2 and interleukin 2 signaling. So now you're no longer needing that co-stimulation with the MHC class 2 antigen presenting cells, professional antigenic cells like dendritic cells. That's lost. You don't need it any longer to co-activate. You also don't need IL-2 anymore. It more depends almost entirely on microbial antigens like pathobiont antigens. So these Tregs subsequently fill up the lamina propria. This is where we are in the gut. Treg compartment. However, whether these can differentiate into Treg subsites is not known. But you're starting to generate a pro-inflammatory condition, right? Because you're moving away from the controlling mechanism of interleukin 2 and of this, because you're generating the CMAF. And so that modifies overall how Treg cell lineage goes. Certain Treg lineage can go directly to actually Th17 lineage, which of course is pro-inflammatory. So a third subtype of T regulatory cells in the gut express GATA3, not ROR gamma T. That's gone now, and constitutes about a third of all the Tregs. Those FOXP3, GATA3 Tregs express ST2 receptor, which we already talked about, which binds a tissue damage-induced alarmin, which is, of course, interleukin 33. So FOXP3, GATA3 Tregs are instrumental in suppressing inflammation, facilitating tissue repair as well by secretion of amphiregulin and tissue damage. So this is all happening simultaneously. So you have three different T regulatory systems. Each one of them, as long as they are kept in the right stoichiometry, relative concentrations to one another, that's what I mean by stoichiometry, borrowing from chemical reactivity, right? Substrates, the stoichiometry of a reaction. It's the cellular stoichiometry. You maintain a healthy lamina propria mucosal lining of the gut environment, a niche. But if you manipulate that, and that's manipulated by altering the influx of, say, short-chain fatty acids, or modifying the secretion and production of certain antigens because of a change in the microflora, you will start modulating this whole T regulatory system. And when you modulate it, you can also defeat some of those T cell lineages and ultimately turn on an inflammatory response. So that's the whole key. Right? See how complicated that is. Now, let's do a little bit of logic. There is something called the traditional square of opposition. Okay? A logical opposition occurs among standard form catechol propositions. For example, if they have the same subject and predicate terms and they differ in quality, quantity. Okay, these are two general sorts of categories, or both. They're components of the modulation of the existence of modifying systems in any kind of natural environment or any kind of physical phenomenological environment, actually. So consider the kinds of opposition that can arise with differing quantity and quality. Case one, differ in quantity and quality. Two, only in quality but not quantity. So this is the quantum versus quale. Differ in quantity, so quantum effects, but not the quality, no quale changes. And then same quantity and quality, and that's a trivial case where nothing's happened. So here's a square of opposition. You have A, E, okay, these are propositions, I, O propositions. A, E, I, O propositions. And you can see that there's relationships between A and E, A and I, A and O, and the same with E to A, E to I, E to O. And there's a relationship between the propositions I to O. Some are contrary, which will allow you to carry out a dialectic, that is thesis, antithesis, synthesis, or they're contradictory, which you can do thesis, antithesis, full stop. You can't synthesize contradictions. So, four corners of the chart represent four basic forms of propositions. A propositions are universal affirmatives. They take the form all SRP. E propositions are universal negations. They take the form of no SRP. See? So they're contrary. I propositions are particular affirmatives. See, they're in this subalternation domain from A. So the particular affirmatives, they are some SRP, and the subalternation from the universal negation goes to O. O propositions are particular negations, take the form of some SR not P. Okay. Now, first, A and O propositions are contradictory, okay, as are E and I propositions. So propositions are contradictory when the truth of one implies the falsity of the other. And conversely, here we see that the truth of a proposition in the form of all S or P implies the falsity of the corresponding proposition in the form some S or not P. See? For example, if the proposition all, here we go now, here's where I'm using the square of opposition. If the proposition all Treg pharmacological modulation is tractable, which means we can follow it, we can monitor it, we therefore may be able to modulate it. It's a universal affirmative, A. If that's true, then the proposition some Treg pharmacological modulation is not tractable, O, must be false. It's a contradiction. See how this works. Similarly, if no Treg pharmacological modulation is tractable, that's your universal negation, that is false, then the proposition some Treg pharmacological modulation is tractable, I, has to be true. Okay. So, there's the contradiction down this pathway. So you understand how this works. There are contradictions and there are contrary statements. So if you think about how experiments are designed and you use a square of opposition, like I'm doing here and filling in for universal affirmatives, universal negations, and then the subalternate particular propositions, you can ask these questions before you ever think about what might happen if you modulate, for example, FOXP3 to promote T regulatory stimulation activation. See? So, secondly, A and E propositions are contrary. Now I'm explaining contrary. Propositions are contrary when they cannot both be true. So the A proposition, all Treg pharmacological modulation is tractable, can't be true at the same time as the corresponding E population, no Treg pharmacological modulation is tractable. But note, where they both can't be true, corresponding A and E propositions, while they are contrary, which is what the definition of contrary means, they're not contradictory. So while they can't both be true, they can both be false. See how well the square of opposition, this logical paradigmatic test that you can do before you ever think about experiments, can be useful before you design the experimental design. And then ultimately way downstream, what do you want to modulate in T-cell metabolism to modify, say, the environment for tumors or the environment for cardiovascular disease or the environment for type 2 diabetes, pro-inflammatory responses in the gut. You get it. Next, I and O propositions, okay, are subcontrary. Propositions are subcontrary when it's impossible for both to be false. Okay? Because promoting Treg activation will simultaneously deactivate Th cell activation. Okay, see, I'm plugging in here. It's false. So promoting Treg activation will not simultaneously deactivate Th cell activation. Has to be true. Okay. Note, however, that it is possible for corresponding I and O propositions both to be true, just like A and E could both be false. It's amazing, isn't it? Just using logic, categorical logic. Okay, so let's check this out. As with, quote, some Treg, my quotes, my statements, some Treg activation decreases Th activation, okay, and some Treg activation does not decrease Th cell activation. Again, I and O propositions are subcontrary, but not contrary and not contradictory. So they both can be true. See how powerful this is, right? Finally, two propositions are said to stand in relation of the subaltern when the truth of the first, the superaltern, implies the truth of the second, the subaltern, but not conversely. So A propositions stand in the subalternation relation with the corresponding I propositions. There. The truth of the A proposition, me plugging in now, all Treg activation imparts inflammatory control. Sounds like a good statement, right? I mean, we know some does, okay? Implies the truth of the proposition that some Treg activation imparts inflammatory control. Sure. But it does not imply the truth of the E proposition, no Treg activation imparts inflammatory control. Of course not. In traditional logic, then, the truth of an A or E proposition implies the truth of the corresponding I or O proposition, respectively. That's how the subalterns work in the square of opposition. Consequently, the falsity of an I or an O proposition implies the falsity of the corresponding A or E. So when the particular is false, the universal must also be false. See, it doesn't work the other way. Very, very clever, this square of opposition. So what's the resonating feature here? The truth of a particular proposition does not imply the truth of the corresponding universal. That's basically what I just said. Okay, so particular proposition is looking at T cell lineages and saying, oh, well, we got this going on with FOXP3 and PPR gamma and ROR gamma T. And so let's pump this out and say that's the same thing that's going to happen in this other cell lineage or in this other tissue type. You can't do that, right? So, nor does the falsity of a universal proposition, for example, that all T regulation leads to anti-inflammation, that doesn't necessarily carry downwards to the respective particular proposition because those are subalterns. So you see how that works? So once you get to an induction and you want to say everything works this way, after you do an experiment, you get your data, you verify your data into evidence, and ultimately you want to say this evidence, you throw that evidence against the largesse and the logos of what is already published in a given area. As you look at the literature, compare and contrast it, right? You do a comparison of what's out there in the entire opus of the literature in that area. And you get an induction, you get a universal perspective, right? It doesn't mean that the falsity, if you find something that it says, no, you can't do this, it doesn't mean that in a particular case it wouldn't work. Right? But you can't go the other way. So it's very, very, very important to use this categorical logic. So here's your existential import I'm trying to get at here. Without the traditional presuppositions of existential import, that is, the supposition that all categories have at least one member, because you don't know that. You can't suppose that because this is biology, right? Then only the contradictory relationships really hold. So on what is sometimes called the modern square of opposition, this was generated by logicians, the lines for contraries, subcontraries, and subalternation are all erased, leaving only the diagonal lines for contradictions. And that's what often happens when you start to look at the fine-grained details in clinical experiments with humans when you're trying to use an immunopharmacological modulating agent. You start finding out that you cannot get there from here, meaning that what you thought was going to function because you saw it before isn't functioning in the specific. Or what you saw in the specific, the deduction, doing the deductive, hypothetical deduction to get to an experimental design also doesn't play a role necessarily universally. Yeah. So if we cannot presuppose and therefore assert as the first premise, because it's never demonstrated, we don't know that everything is exactly right? That we have, for example, at least one member in every one of those categories, all SRP, no SRP, some SRP, some SRP, those categories, okay? So the concept that full, here I'm plugging in again, Treg activation is necessary for control over inflammation, then we cannot assert that some Treg activation is necessary for a control over inflammation. So just using the square of opposition. And this is indeed what we find if we look at the literature. Therefore, we can only say that Treg activation may or may not control inflammation. It's really all you can say from a logical perspective, categorical logic. So ultimately, it translates to a hypothetical deduction, going back to the beginning of experimental designs. Clinically, remember, a hypothesis, a hypothesis is below a thesis, right? Hypo is below. Clinically controlling T-cell mediated acquired immunity may never resolve satisfactorily to become standard medical practice. That's what logically the conclusion you can come to. Before you ever try to design, given, say, antibodies to specific transcription factors or cytokines or growth factors, or regulating short-chain fatty acid metabolism, or regulating mevalonic acid metabolism, or beta oxidation, or glycolysis, the enolases, any of that, you're never going to be able to say that it's always going to work, okay? And in fact, even considering controlling T-cell mediated acquired immunity in the 30,000 foot level may not be the way to go. So how do you remedy this, right? How do you don't stop doing experiments, you don't stop trying. My suggestion, which I will develop later in a subsequent discussion, subsequent podcasts, rather than looking at mutants, rather than looking at cells, rather than looking ever at any kind of animal models exclusively and then extrapolating up to humans, every time there is any kind of surgery, get tissue and keep that tissue viable and do a complete analysis of that tissue at the level of lipids, carbohydrates, nucleic acids, proteins, transcripts, and get tissue from healthy subjects. If we know how the healthy system works, we really know how it's regulated. Because when we're looking at a pathological state, when one thing goes wrong, 30 things can go wrong. And maybe 10 don't. But in one system, 10 won't, but 5 will. In another system, 5 will, but 10 won't. See? Start with the healthy system and try to understand the overall global regulation by getting enough data at that level. So rather than doing machine learning, which is saying this is what the data says, let's move on, be intelligent at the beginning using things like logic and take a look at healthy tissue and try to understand the coordinated regulation of all these cofactors, transcription factors, kinase cascades, gene expression systems, the whole profile of antigen-mediated induction of T cells and B cells and whatnot, before we start looking at pathobiological systems. And then look at how that changes by subtraction in different pathological states. And then look at the genome and epigenome of each of those people. QED. So the dialectical event-based ontological record. If there is a phenomenological distinction between the mind and the body, there would be a physical-chemical axis of interaction. Okay? So now I'm reaching way out and showing you how I got to using this in biomedicine. That axis might be the molecular and cellular adaptive immunological interactome, which is what we've been talking about all these last episodes, that serves to generate neural tracts according to developmental, endocrine, and peripheral stimuli while maintaining repair processes. This is like overall the healthy environment, right? The physiological environment in the central nervous system by using the complex interactions among oligodendrocytes, astrocytes, microglia, and neurons. So I'm trying to get to a global understanding of the immune modulation. It doesn't only work in pathological states or in surveillance or in controlling antigens. My argument is that it's there all the time during development and differentiation and maintenance of the central nervous system. And the central nervous system actually is an immune-based system. And the word immune has to be replaced now with a new term. And I'm suggesting Diaeventontological systems, right? Dialectical event ontologies rather than substance ontologies. That way you get away from any specific subset of biochemistry or molecular genetics. Now, according to my view, the neural networks are processes, again, process type of biology, event ontology, becoming eventual in nature This contrasts this with substance ontology, like how much of STAT3, how much of FOXP3. That's substance ontology, like what's there and how much, right? Quale, quantity, quale, quantum, rather than looking at how they change over time in healthy systems. So, the sequence and development pattern of modifiable events appears energetic waveform fields rather than particles of matter, like chemical bonds do. An event ontology that reaches across the neural networks of genetic and environmental interactions is articulated and synthesized through an epigenetic modification, which we see all the time for gene expression, of the classical immune system. It's always available, it's always there, it's always interacting with every biological situation, thus creating a responsive plastic and elastic memory field capable of learning, ideation, imagination, and understanding through time. So that's, I believe, how the brain actually changes over time in any individual. And of course, with the brain, all of the components of the central nervous system, which are autoregulatory primarily in disease state. So this dialectical, which involves thesis, antithesis, synthesis, right? As long as you can stay out of contradictions, the event ontology involves a central existing individual agentically interacting and interfacing through the present perfect just happened processes obtained across the inherited genome, the changing environment, and the transcendental, that's like above it all, the regulation above it all, of an immunoepigenome through time. And there is what I mean by a diaventome. You have the genome, you have the environment, and you have the immunoepigenome. Neural networks are processes, central individual agentically interacting through events through time. Diaeventontology, again, dialectical event ontology, can be deployed as an adaptational, processive, scientific accounting of the healthy physiological human condition, through a biochemical and cellular event ontology. What's there? How does it change over time? Its biological mechanism of action, linking immunophysiological states like T regulatory cell activation and acquired immunity, incorporates the immunoepigenomic induction, neural, endocrine, and metabolic response. We just saw that, adipose tissue, GALT, right? The entire immune system, lymphatic system, right? And the micro and macro environment, microorganisms, as well as what cell type you're talking about, what tissue type, the signaling across those tissues, the extracellular matrix, phosphorylation cascades, secretion of proteins, degradation of proteins, turnover of RNAs, splicing of RNAs, right? Through classical constitutive surveillance, it's all happening inside immune cells, and acquired effector cellular humoral defenses, see T cells, B cells, for example. Stratagems utilizing reversible covalent modification and hydrophobic interactions of nucleic acids, proteins, and lipids. That's my global understanding of what's going on inside the living system. Diaeventontology offers a general molecular system theory for a free will-driven, agency-based individual adaptation and a knowledge-acquiring physiological rational mechanism that better explains the core event ontology of human existence, including health and well-being. An experimental strategy that exclusively studies the acquired immune response in healthy individuals through long-term temporal and spatial tracking and surveillance of extra and intracellular lymphocyte differentiation and interconversion during microbial, genetic, and abiotic stress should, or I'm saying will, generate a phenomenological event ontology that succeeds to varying degrees the disease-free survival you see in the living human body until such times that things go wrong and that body dies. Right? Okay. So I put that all together here for the Tregs. Genome, I'm only putting a small subset of genes, FOXP3, FOXO13, TGF-beta, the NIFL3 we talked about, which has to go down in Treg cells. I talked about that in the Authentic Biochemistry lectures. Interleukin-2, the SMAD system, the Mtor system, the TCR, T-cell receptor, the ROR gamma T. Environment, we did not talk about it today, but I have another VerEvMed and Authentic Biochemistry lectures. Aging has an effect on all this. Gender has an effect on this. That actually has to do with estrogen and testosterone mediating immune responses. That's well established, actually. Inflammation itself. Cardiovascular disease is part of the environment. Infection, stress, oncogenesis, or lack thereof. That is oncogenesis that fails, oncogenesis that becomes metastatic, for example. Sedentary lifestyle plays a role. Obesity is an overarching environmental condition in the increasingly overweight population in the United States and worldwide. Visceral adipose in particular can be looked at as a subset of global obesity. Hematopoiesis is important in terms of what cell lineages are being made. The digestive system, we didn't even really scratch the surface today, but we have in the past, and we will again. And the Gulf, which is part of the digestive system. So what about the immunoepigenome here? Again, this is a trigonal planar system with Treg in the middle. Macrophage switching, we talked about M1, M2. CpG methylation, demethylation, right? The enzymes involved in that. Histone acetylation, deacetylation, the sirtuins, right? B and plasma cell recruitment. T cell differentiation and modulation, something we've been hitting hard on these last several lectures. And of course, cytokine, chemokine, growth factor modulation, and the microRNA, which is all part of the epigenome. So we talked about the universal affirmatives, universal negatives, particular affirmatives, and particular negatives. All of this then has to be taken into account just when we're thinking about Treg Diaeventontologics. So every time we think about what we want to modulate in a given system to be able to move forward in understanding pathological states, my recommendation is we start with categorical logic. We look at what's published very carefully. We design experiments around that largesse of information that has been gathered from thousands and thousands of experiments, all of which together will give us a synthesis that can be plugged into a Diaeventontological framework to move on, to do that hypothetical deduction, design experiments to look at the pathological state with a more logical parameter, rather than shooting in the dark or using something which I think is absurd like machine learning. So that's it for today. Again, that's me, Dr. Dan Guerra. That's my email address. There's the email address for VerEvMed, the website for VerEvMed. VerEvMed means scientists verifying published evidence in medical biosciences. We take on clients and we look at we target specific research areas, work with you, explore it, dissect it, disambiguate it, and send back to you what we have found based on your level of credentialing, whether or not you're a clinician, you're a theorist, you're a systematicist, you're a graduate student, you're a layperson. We can look at the literature and we can gear it to your level of understanding the terminology so that we can help you, whoever you happen to be, to look at authentic scientific information and be able to dissect it logically and carefully so that you really know what science is saying, rather than what's been synthesized for you and regurgitated by people who are often not even scientists. Okay? So that's not what we are. We're scientists. And that's how we function here. Okay, so let's finish up. All right, so what I want to say, like I always do at the end of all of these talks, is my sign-off, and that is, it was a great pleasure to give you this lecture. Hopefully you gained something from it. Please respond to it. Please respond by joining my, going to my Facebook page, liking my Facebook page, looking at my VerEvMed lectures on YouTube and liking them and asking to join, and also by looking at my Authentic Biochemistry podcast. It's called Authentic Biochemistry. Look at that, and also please subscribe to that, because we really would like more and more people to follow our work so that we can do more of this and we can be very specific as to what you want me to discuss. And I'm at your service all the time, and all I'm saying is bye for now.